Welcome back. So today we will discuss about the urine formation, and in urine formation we will see that how the filtrate are coming out from the blood and filtrating out from the Bowman capsule is converting into what we call urine. So we will discuss this whole formation of urine in details. We will also try to cover some minute details which is very important in order to understand the whole phenomena of urine formation and some of the terminologies which are associated with this urine formation. So if we go to the slide, these are the three steps in urine formation. So the first step is glomerular filtration, second is reabsorption, and the third is secretion. So these are the three steps in the urine formation, and we will try to discuss each and every step in detail, and some of the minute stuff related to the urine formation individually we will try to cover. Now in the first step, the glomerular filtration itself indicates that something is filtrating out from the blood. Now this whole phenomena is very important in maintaining the fluid balance in the body. So the body fluid balance is largely depend upon all these three processes whatsoever we discussed just now. So body needs to synchronize, or kidney particularly needs to synchronize these three processes in a very nice way, so that to get the urine in actual concentration. On an average 180 liters of blood is getting filtered. Now out of 180 liters of filtrate, what the body is producing urine is approximately 1.5 liters. So 1.5 liters is the actual amount of the urine that is excreted out from the body every day. Now if you see the proportion, approximately 99% of the stuffs are getting back into the body, because these are very important. Suppose this is an afferent duct. Now we will try to skip the word duct, we will say arteriole, because now we are discussing about the blood flow. So the blood flow is coming from into this glomerular by this arterioles, and this arterioles is called afferent arteriole. And similarly the blood is coming out after passing through this capillary which we call glomerulus, and the blood which is passing out is called efferent arteriole. So efferent arteriole and afferent arteriole are the one which is carrying the blood and all. Now what is happening here, the blood is coming and getting passed through this small like meshwork which we are calling glomerulus, and then we have this cup like structure, we are calling it Bowman capsule, Bowman's capsule. And then what we discussed in the last class, that we have this finger like projection called podocyte, and this podocyte is having some holes. So we have some holes here, and these holes are nothing but these are called slits, and otherwise we can simply call it, sometimes these are also called as fenestrera. So let's see, we can just write down here. So these holes which are called as slits are also called fenestrera. The blood filtrate is coming out. Now what is coming out is the complete fluid part except the cellular portion, the RBC and protein. So proteins are not coming out. So this fluid which is coming out from this filtrate is free of RBC, the cellular material, and protein. So these are protein free, cellular free material that is coming out in Bowman capsule, and from Bowman capsule this is passing into this tube. Every day the kidney is filtrating approximately 180 liters of the blood. Now out of 180 liters of the blood, only 1.5 is coming out. So what is coming out is 1.5. What about the other? The amount 178.5 liters of the content is getting reabsorbed into the circulatory system or the capillary network which is running parallel to this thing. So there is a reabsorption process here. So things are getting reabsorbed, and to maintain the ionic balance or to maintain the ionic equilibrium of the fluid, something is secreted into the duct also. So there is secretion. So there is secretion of certain kind of ions into this duct or this tube that is making the osmoregulation, the balance of the ion, the pH maintenance and everything, and that is causing this whole phenomena to be sequestered in 3 distinct phases. Filtration, this is the first process. Second is reabsorption, that means the whole fluid which is coming into this filtrate is not passing out. So like complete 99% of the things are getting reabsorbed, and to maintain the ionic balance there is some secretion coming into the duct to maintain the osmoregulation. So this is the 3 distinct processes which we are going to discuss one by one in detail. So the first thing is we need to understand that the blood is coming from the afferent arteriole, and that is having certain pressure. The blood pressure is there, and because of this pressure only the things are getting filtered out, and this pressure is called glomerular filtration rate. So glomerular filtration rate, GFR. So let's see. So we will now discuss the one very important process of filtration, and then we will see that how this filtrate is getting reabsorbed and getting some secretion from the other side. So what we are discussing is glomerular filtration rate. In short, it is called glomerular filtration, GFR. So glomerular filtration rate in human being, in a normal healthy person, is approximately 125 ml per minute. So on an average, if you multiply the complete day count is coming approximately 180 liter of the blood is kind of filtrating every day through this blood pressure on the afferent arteriole side. Blood pressure is maintained by certain kind of secretion or certain kind of chemicals secreted in certain areas in this nephrons. So we will try to discuss some very important areas in this nephron regulating the blood pressure, ultimately regulating the glomerular filtration rate. So let's see those areas. So what is happening here, that the whole glomerular as such, we will try to draw the glomerulus one more time. So we have this, so let's see, this is our Bowman capsule, and then we have this proximal convoluted tubules, and then we have this lobes of Henle, and then we have this, we will draw this glomerular afferent, and then we have this efferent duct. So this is afferent arterioles, efferent arterioles, then we have glomerulus, I am just writing the short form, then we have this proximal convoluted tubules, this is Henle's loop or Henle's loop, and then this distal convoluted tubule is kind of passing through this area. So this distal convoluted tubule, which is called DCT, distal convoluted tubule. Now one should remember this part, that this whole is the part of cortex and this whole is a part of medulla. Now this is the medullary portion and this is the cortical portion. And if you don't have the Henel's kind of a structure in this nephron, you can call it cortical nephrons. Otherwise, you can call it juxtamedullary nephrons. Now, these nephrons here are having distal convoluted tubules crossing very close to this afferent and efferent arteriole. Now, this passage is very important. Now, because of this closeness or proximity of the blood vessels, there is a very unique phenomena going on in this area. So, these cells in the distal convoluted tubules are getting modified in order to do some process. And this modification, this area is very important, and this whole area is called macula densa. Macula densa. M-A-C-U-L-A densa. So, macula densa is just an area in the distal convoluted tubule. Remember, these are the modification of the cell in distal convoluted tubule passing very close to afferent arteriole and efferent arteriole, and these modifications are there in order to do certain kind of modulation or controlling the blood pressure coming from this area. Now, here we have very specialized cell. Now, we will try to see this area. So, let us see, here we have this Bowman capsule, and then we have this glomerulus, and this is afferent, this is efferent, and then we have this, here the cells are called macula densa. Now, the cells in the afferent arteriole and the efferent arterioles are getting modified. So, we have called afferent arteriole and efferent arteriole. So, this is a little bit lengthy, but one has to understand that what's going on in this area. So, here the cells are getting modified. Now, this modification is same just to control the blood pressure, because this is very, very important phenomena. The blood is coming and passing all the stuff in downstream. So, if you can't control the blood pressure, the things will not maintain properly. So, here you have certain kind of modification. So, the cells in this close proximity to the glomerulus. So, the cells in the afferent arteriole, efferent arterioles are getting modified to do certain things. And these modified cells are called juxtaglomerular cell. Juxtaglomerular cell. Juxtaglomerular cells. Now, this juxta means close. Juxta means just vicinity. So, closeness with the glomerulus. So, these cells are very close to this glomerulus, this capillary network. That is why these are called as juxtaglomerular cell, and this whole area is called juxtaglomerular apparatus. So, juxtaglomerular cells are very important because they used to secrete an enzyme called renin. So, let us see. So, we understood this macula densa, and now we are trying to see why this area is important. So, this whole cellular part, the juxtaglomerular cell or juxtaglomerular apparatus, simply sometimes they are called as juxtaglomerular apparatus, is very important, and these cells are modified to secrete a very specific enzyme, and that enzyme is called renin. Now, renin is a very unique enzyme secreted by juxtaglomerular cells in this area where the afferent and efferent arterioles are there in order to maintain the blood pressure, in order to maintain the blood flow, and this is very important enzyme. This should not be confused with another enzyme that is secreted in the stomach. So, there is another enzyme called renin, and this renin is double anion. Renin is an enzyme secreted in the bovine stomach and used to make cheese. So this is very important commercial enzyme that is utilized in the manufacture of cheese, and that is isolated from the bovine stomach, and the enzyme is called renin. This should not be confused with this renin which is secreted by juxtaglomerular cell in the area of nephron, in the area of afferent and efferent arteriole. These are the modified cells in order to control the blood flow towards the glomerular area. So now we have this whole filtrate coming out. Now this filtrate, whatsoever is coming out, is having all the nutrients. So what we discuss is filtration. So thus filtration is over. So here we have again, I am drawing it again and again in order to give proper understanding. Afferent arteriole, then these are glomerular, and then these are the tubes starting of this convoluted tubule, the proximal part of the tubule. Now what will happen? Now here the things are coming, all the content except protein and the cellular part. So here we have complete blood. Now what we have here is blood minus cellular debris or cellular particle minus and all the, so we don't have cells and we don't have the protein part, the amino acid and the protein content in the blood. So the protein content is also not here. So whatever is coming here should be absorbed, because these are very important ingredient for the human body. So lot of things are absorbed here. So let's draw in clear diagrammatic representation. Again we will draw here. So we have cortex and then we have medulla. So what is happening here, that this proximal convoluted tubule, so this proximal convoluted tubules is continuing into this Hanel's loop. So this Hanel's loop is little bit thicker in the upper area in the beginning, and then it is getting tapered, and this tapered is also here, and then in the upper part it is getting a little bit of the thicker nature, and then this is kind of continuing into this distal convoluted tubule, and of course this final is coming in the form of, we have this collecting duct, So here what we are now going to discuss is how the things are getting reabsorbed. Now we know that the major concentration of the filtrate is water. So major concentration, 70 to 80% of the complete water is getting absorbed in this area. So complete water, we will see one by one. Complete water is getting reabsorbed into the body system. In order to avoid any water loss, about 80%, 70 to 80% of the water is getting absorbed from the proximal convoluted tubule. Remember, this part is very important. Proximal convoluted tubule is absorbing 70 to 80% of water. Remaining part of the water is absorbed through this particular portion, and here and here. So if I draw the complete picture, so the water is absorbed in the proximal convoluted tubule, Henle loop in the descending loop, not in the ascending loop. In the ascending loop, there is no water absorption. This is very important to remember, that the ascending loop of Henle's or Henle's ascending loop is impermeable to water. So water is not getting any kind of movement or any kind of fluctuation in this particular portion. While distal convoluted tubule is also absorbing or also help in reabsorbing water as such, and rest all are collected through the collecting duct, so that to finally create what we call urine. We have ions, and some of the ions which are necessary for the body is NaCl. So NaCl is getting reabsorbed here in the proximal convoluted tubule and also from the distal convoluted tubules. So these ions are very important. While our ascending loop, which is impermeable to water, is permeable to salt. So lot of salts are getting reabsorbed from the distal part, or the ascending loop of Henle's. We are having lot of absorption of these ions, the sodium ions. Apart from the sodium ions, we have bicarbonate ion. So we have here reabsorption of SCO3 minus ion also, and also from distal convoluted tubules. So we will see here that what is happening, that most of the things are getting reabsorbed from the proximal convoluted tubules and some part to distal convoluted tubules. While the descending loop of Henle's is involved in water absorption, while ascending loop is involved in ions absorption, particularly sodium chloride, sodium ions. Now we will now finish this absorption part. We will jump into the third part, secretion, and in the secretion we would like to discuss that what are the secretion involved in order to maintain the balance, and this balance is coming by some of the ions secreted into the tubes to make the pH constant, to make the ionic equilibrium constant, and those two very important ions are H plus ion and potassium ion. So my arrow will go like this. So H plus ion and potassium ion is coming into the ductal part, and also you have another ion, and that is ammonia. So ammonia ion is also coming into this ductal part. So these are the secretion, into the system in order to make the fluid balance constant in this area. Also in this distal convoluted tubule, we have H plus and K plus ion moving into the system, so that the whole process of secretion is synchronized. Now if we start with the proximal convoluted tubules, the second part is Henle's loop, third is distal convoluted tubules, and the fourth is collecting duct. So these are the four ductal system, and mostly the proximal convoluted tubules are involved in absorbing water, sodium chloride and bicarbonate ion, and there is secretion of the two more important ions, which is K plus and H plus ion, and in addition we have ammonia. And in Henle's loop we have absorption of water from the descending limb, and from the ascending limb absorption of NaCl. Similarly, from distal convoluted tubule we have water absorption, we have NaCl absorption, we have bicarbonate absorption, and then we have K plus and H plus ion secretions in the distal convoluted part. In the collecting duct we have water absorption, and secretion wise we have some other secretion which are important to maintain the osmoregulation of the collecting duct. So, but these are in total about the major contribution of this tubular system in the nephron in kidney. Now there are two things which I need to discuss here. The first thing is about this cells, very important cells in the proximal convoluted tubule. So let's see the cells in the proximal convoluted tubule, and second thing we will discuss about the renin, the enzyme which is secreted by juxtaglomerular cells. So this proximal convoluted tubules, if we divide the cellular system in the human body, the cellular system in the human body is grossly divided into 3 distinct types. So the first type, if we see, because I just wanted to give an idea, it's not important here, but just to get an idea that what are the cells involved in different body area. We have the epithelium. So we have certain kind of cells which are very flat in nature. So we have cells which are very flat. So if you see the cells like this, you can see the nucleus coming out from the cell like an omelette. So you have those cells having nucleus bulge out from the surface. So particularly the cell will look like this. So the nucleus is kind of bulge out. This kind of cellular arrangement in the epithelium is called squamous epithelium. Squamous epithelium. The second kind of cells are roughly cubical in nature. So we have this rough cubical arrangement of the cells, and the nucleus is in the center. So these kinds of cells are called cuboidal because they are like cubes, so cuboidal. The proximal convoluted tubule cells are cuboidal epithelium. They are breast border cuboidal epithelium. They are very important for secretion. So because we are talking about secretion, lot of things are coming, and secretion, wherever secretions are involved in the human body, the lining of those glands, the lining of those body cavities are lined by cuboidal epithelium. Remember, these cuboidal epitheliums are involved in the area where you have secretion coming up. So secretion is there in the proximal convoluted tubule, so that's why the epithelium is cuboidal epithelium. Now, what we discuss in the kidney, we have in the last portion where the urines are collected out, is called urinary bladder, and there urinary bladder we have a very specific cell, what we discussed earlier is called transitional epithelium, that is having flexibility to expand or relax or contract according to the volume of the fluid inside urinary bladder. Apart from cuboidal epithelium, we have small column of cell. So the cells are elongated, bigger inside, and the nucleus is in the bottom. These are called columnar epithelium. Columnar epithelium. So the columnar epithelium, cuboidal epithelium, and squamous epithelium are the 3 distinct types of epithelial cells based on the shape. The columnar epithelium, because these are column-like cells, these are available in the gastrointestinal cavity, the stomach, the small intestine. These are the cells which are lining the small intestine, help in the absorption of the food particle and all. But these are the cells which are involved in secretion. So the glands and the area here in proximal convoluted tubules are having cuboidal epithelium. So just for your knowledge, these are the 3 kinds of distinct cells. So this is all about the proximal convoluted tubules and the cells involved in this proximal convoluted tubule. Now, we will discuss about this very important enzyme renin secreted by juxtaglomerular cells or the juxtaglomerular apparatus, which are the cells modified very close to the glomerulus in order to maintain the blood pressure. Now, what is happening? This renin is converting, the enzyme renin is important in converting angiotensinogen into angiotensin 1. So there is another protein, kind of a very important regulator of blood pressure, and that is called angiotensin. Now this angiotensin, as name itself indicates, tensin means tension, angio means blood vessel. So we have this blood vessels. If you say formation of blood vessels, we call it angiogenesis. Angiogenesis is nothing but the formation of blood vessels. So here angiotensin is nothing but that can give tension to the blood vessel. That means if you dilate the blood vessel, the blood flow will decrease. That means the pressure. If you dilate the blood vessel, the pressure will decrease. If you constrict the blood vessels, the pressure will increase. So this dilation and contraction is governed by this particular kind of thing. So angiotensin is in the precursor form. We called it angiotensinogen. So angiotensin is the precursor of another form which is called angiotensin. Angiotensin 1, which is converted into angiotensin 2. Now this angiotensinogen conversion into angiotensin 1 is done by this enzyme, and that is called renin. So this angiotensin 2 is what is causing increase in the blood pressure. How? Suppose if, now this is, suppose we are just drawing a glomerular area here and the blood is flowing here in order to do filtration. If the blood flow is slow, if the blood flow towards the glomerulus is slow, if the blood flow is not appropriate, the filtration process will not be appropriate. If filtration process is not appropriate, the waste material, the unwanted material will not be filtered out through this kidney, and because of this reason the toxicity will start in the body area. So this maintenance of this pressure is very important. Now this angiotensinogen conversion to angiotensin 1 and 2 further in presence of renin is causing the vasoconstriction. So the arteriole will cause a vasoconstriction. So because of constriction, so what will happen? Suppose you have the blood vessel like this will constrict. So the diameter of this blood vessel will decrease, and this process is called vasoconstriction. So we will write here. The process is called vasoconstriction. Vaso means vessel, constriction means constriction, the decrease in the diameter, and because of this reason the blood pressure will increase, and if the blood pressure will increase, more and more blood will pass on, right, and because of this reason the filtration rate will be fast. What we have discussed earlier, glomerular filtration rate will increase, right. So the whole phenomena is synchronized in such a way. If the blood pressure is less, the body, the nephron, the whole system that's controlled by neurons, we will see it later on again when we will discuss the regulation of the urine formation, regulation of the nephron, regulation of the kidney and all. But here, since we discussed about the renin, we need to understand that once the pressure is decreased or pressure is down towards this area, the filtration will not happen. And now we know that this particular area is having this modified cell which is called JG cell, and this juxtaglomerular cell is sensing this low pressure, and because of this sense they are secreting this particular kind of enzyme which is converting this angiotensinogen into angiotensin 1 and 2, that is causing vasoconstriction, and because of vasoconstriction the flow is increasing, glomerular filtration rate is increasing. So these are kind of feedback mechanism. Body homeostasis mechanism is very much synchronized. So if there is upregulation of one thing, there is a counter mechanism available in the body to nullify that thing. So things should be in the equilibrium. One should not go in a very high concentration, one should not go in a very low concentration. So things should be very much constant. So if a renin concentration is high, that will cause more and more production of angiotensin 2, and the whole process will cause more glomerular filtration. So more and more urine formation. Now, if there is more and more urine formation, suppose body don't have the much, body wants to retain more and more, they need to counteract this phenomena. Otherwise, there will be loss of water, more loss of water and all. To reduce this, there should be a feedback mechanism, negative feedback. So, the neuron will sense all those things and give a negative feedback so as to stop the renin production. So, these are all positive feedback and negative feedback phenomena to control renin. So, this renin is very, very important in order to do blood pressure maintenance. That was we discussed today, urine formation and all. This whole renin and angiotensin is very unique system available in nephrons. This is called Renin-Angiotensin System. So, this is called Renin-Angiotensin System. So, this Renin-Angiotensin System is a very unique system available in nephrons to control blood pressure by converting angiotensinogen to angiotensin 2 in presence of this renin enzyme that is causing ultimate vasoconstriction and the modulation of blood pressure. So, to summarize the complete urine formation, let us go and individually see in the slide form so that you guys have a better understanding about the urine formation. So, here is the complete diagrammatic representation of the nephrons. So, we have this inlet where we have this afferent arteriole, and then in the first here the blood is coming, and from here the blood is leaving after filtration. So, the first process here is filtration, and in this filtration we will just say this is an ultrafiltration, because what we know that everything is coming out except the proteins and the RBC, the blood cells. So, that is why we can just simply call it ultrafiltration, and from there this filtrate is passing downstream, and in the second, this second number is what indicates for reabsorption, while the third one is what we have some kind of secretions coming from the ductal side, and this is nothing, this area is nothing but the capillary network. So, these are nothing but the capillary network. So, this whole capillary network is having the blood connection, and we will see later on what is the modified version of this capillary, what we call as vasa recta. So, this vasa recta is nothing but this capillary network which is which are running parallel to the Henle's loop, and here we have this renal vein. So, here we have this blood picture, and the last one after filtrate what we are getting here is urine. So, this one is complete blood side. So, we can simply call this is blood, and here we can simply call it the urine. So, there is a complete formation of urine by this ultrafiltration method, and that is what we discussed completely here, and this is for better understanding in diagrammatic wise. Next, we discuss about this unique cells which are located in the afferent arteriole and efferent arteriole, and they are very close to glomerulus. That is why they are called as JG cell or cells, or they are simply called as Juxtaglomerular cells, and these cells are here. So, these are some of the modified cells which are very much defined to secrete a very specific kind of enzyme, and what we discussed, that enzyme is renin. So, this renin-angiotensin system is what is comprising comprises by this kind of cellular architect around nephrons, and this the circular vessel is nothing, is look like vessel, but this is duct, and this is the part of distal convoluted tubule, and this distal convoluted tubule is nothing but macula densa. So, this macula densa is kind of a modified distal convoluted tubule, and this juxtaglomerular cells is what is called the angiotensin system, and of course, these are all the Bowman capsule area, Bowman's capsule. This is proximal convoluted tubule, and this we simply call glomerulus. And here these are the projection, that is what we discussed. These are podocyte, and these are nothing. Podocytes are the cells which are kind of having this pore which we call slits or simply fenestrae. These holes are actually giving filtered to the urine which is coming here, a divide of any cellular fragment and amino acid. So, what is filtered out is going to be reabsorbed. So, this is about the location of juxtaglomerular cells which are forming this juxtaglomerular apparatus, which is very important to maintain the blood flow in and around these nephrons. Last, we discussed about the actual flow or actual absorption biology or absorption parameter of our nephron. So, here the nephron is, so this is what we have discussed, these are proximal convoluted tubule. So, after this proximal convoluted tubule, we have this area that is called Henle's loop, and here the top portion of the Henle's loop is somewhat thick, while the bottom area is kind of thin. And here we can simply call this is descending limb and this is ascending limb. Here this is ascending limb and this is descending limb of Henle's. And here is the area that is what we discuss, macula densa, which is kind of a modification of the distal convoluted tubule. So, this is DCT, and this is the final area which we are calling collecting duct. So, I am repeating it many times in order to give you better understanding, so that you should not miss any questions coming out of this. So, basically the blood is filtering here. So, this is going in this direction, crossing the proximal convoluted tubule, and this is coming in this descending loops of Henle, and then it is crossing through this ascending limb and finally going through this macula densa, crossing this distal convoluted tubule and finally collecting into this collecting duct, and ultimately this urine is passing through the ureter and finally to urethra. Now, what we discussed here, that this particular area is giving reabsorption to water, and this particular area is giving reabsorption to salts, and then finally this collecting duct is also giving absorption, reabsorption to water. One thing which I forgot there during the explanation of this absorption, that certain kind of urea is also kind of reabsorbed from the collecting duct, and little bit of urea is also getting absorbed in the ascending limbs of ascending part of Henle's loop. So, this urea is slightly getting absorbed here in the ascending part of Henle's loop, while what we discussed here in the proximal convoluted tubule, we have absorption of all water, and then we have salt, and then we have bicarbonate ions, while we have secretion of so there are secretions here, and this secretion are mainly comprises of H plus, K plus and ammonia. So, these are the three actual ionic concentration coming or secreted into the system, while most of the stuffs are getting reabsorbed. So, this is in a nutshell about the complete urine formation, and once we have gone through this urine formation, now in the next lecture what we will discuss is how this urine formation is controlled, how this mechanism of urine formation and how this osmoregularity is maintained, particularly in the Henle's loop along with the concurrent flow of vasa recta. So, we will see very interesting kind of phenomena, how these are happening in that particular area, and how this urine formation, everything is controlled. And in addition, we will also discuss about certain disease condition associated with the urinary system, particularly this excretory aspect of urea and other kind of related accumulation of some toxic material in our next lecture.